Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Advanced Therapy, Safety Management, Proactive Strategies for Real-World Risk Detection. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers who are all from Elucent. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Elucent, who developed the content for this presentation. Elucent is on a mission to help bring new therapies to light by solving the distinct challenges of small and mid-sized biotech companies. The company is purpose-built through the convergence of leading providers to address this unmet need. Today, Elucent is a global provider of comprehensive drug development solutions, including clinical trial operations, regulatory strategy, clinical pharmacology, pharmacovigilance, and biostatistical programming consulting. With more than 30 years of experience in over 70 countries, Elucent's individualized partnership approach provides experience-driven insights and expertise to assist its clients in successfully navigating the complexities of delivering novel treatments to patients. Now, I'd like to welcome our speakers to today's event. And first, I'd like to welcome Dr. Ian Kovacs. And there he is. Hello there, Ian. He has more than 25 years of professional experience in all areas of pharmacovigilance and medical information gained within both the pharmaceutical companies and CROs. In his current position as Global Head of Pharmacovigilance at Elucent, he is responsible for providing strategic leadership and oversight of global pharmacovigilance services provided by Elucent. And next, I'd like to welcome Stephanie Stemp. She is a project director specializing in cell and gene therapy. There she is. She has over 15 years of clinical research experience. She oversees clinical trials, ensuring client satisfaction, timelines, and budgets while managing technical and operational aspects. Beginning in oncology at the site level, she advanced to clinical trial lead and has worked in project management since 2012. And next, I'd like to welcome Dr. Oksana Fabry. She's a Senior Medical Director, Global Head, Hematology and Oncology, and there she is. Hello. Oksana has over 16 years of experience in hematology, oncology, and stem cell transplantation. She is a recognized expert in the development of advanced cell therapies. She brings deep clinical and strategic insight into CAR T cell therapy with a particular focus on overcoming treatment limitations and accelerating innovations. And next is Georgia. There she is, Georgia Kalanji. And Georgia is a health sciences professional with over 16 years of experience in pharmacovigilance. And there she is. Hi, Georgia. Throughout her career, she has developed extensive expertise across all areas of pharmacovigilance and medical information, both from an operational and strategic leadership perspective. In her current role as Associate Director, she is responsible for overseeing post-marketing safety activities, including the development and maintenance of Elucent's local pharmacovigilance network. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to Oksana. So, Oksana, when you're ready, you may begin.
Thank you, Sonia. My name is Oksana Fabry, and I will talk about the clinical aspects of the advanced therapy management. Can we go to the next slide? Advanced therapies are, or as they are defined by the regulatory authorities as advanced therapy medicinal products, are medicines that based on genes, cells, or tissues with the aim to regenerate, repair, or replace damaged tissue or modulate immune responses. These innovative treatments use biological mechanisms or introduce novel genetic material to address fundamental disease causes. They include multiple products, including the gene therapy, such as CAR T cells, gene-modified T cell receptors, and the hematopoietic stem cell gene therapies, as well as T-cell engagers and mRNA-based platforms. What is remarkable is their potential to deliver durable remissions in settings where the standard therapies have failed. But with these breakthroughs comes complexity, particularly around safety. Next slide, please. The mechanisms that drives efficacy also supports safety risks, necessitating balanced innovation and vigilance. Starting from the manufacturing process, where a complex process can introduce batch variability and possible contamination risk. After the cell therapy, we see immune-mediated toxicities like cytokine release syndrome and neurotoxicity, as well as rare events such as hemophagocytic lymphohistiocytosis or tumor lysis syndrome or other rare but potentially dangerous endothelial complications. Because these cells persist, long-term issues like cytopenias and secondary malignancies may emerge months or years later. Unlike small molecule drugs, advanced therapies can lead to persistent biological changes, delayed adverse effects, and require extensive monitoring period. This is why we must plan for safety monitoring from the very first dose all the way through the long-term follow-up. Next slide, please. Safety signals. Safety signals identified during the clinical trials are critical for establishing early risk management strategies, but their manifestation in real-world population may differ significantly, and we will see it in the next slides. In the clinical trial, we see short or immediate risks like cytokine release syndrome, which occurs in approximately 50 to 90% of CAR T recipients, with severe grade cases seen in 10 to 20% of patients. The onset typically is within 14 days post-infusion, with median onset at around day 2 or 3. And we know that tocilizumab and corticosteroids are the mainstay treatments, reducing mortality from more than 15% historically to less than 1% in recent clinical trials. The same is for immune-associated, immune cell-mediated neurotoxicity syndrome. The incidence ranges from 20 to 60% of CAR T cell recipients, with approximately up to 30% patients experiencing severe grade. We know that risk factors include high tumor burden, high CAR T cell dose, and pre-existing neurological conditions. The pathophysiology involves blood-brain barrier disruption and CNS inflammation. We also see another acute reaction, such as infusion-related reaction, which, unlike cytokine release syndrome, these reactions typically occur within minutes or even hours of the administration and are managed with standard supportive care. Also, we see the hepatotoxicity as well in the products, genetically modified products with adenovirus viral gene therapy. And this typically occurs 4 to 8 weeks post-administration. Regarding long-term risk, they are particularly concerning because clinical trials typically have limited follow-up compared to potential life persistence or of genetic modifications. Insertional mutagenesis was observed in early retroviral vector trials in severe combined immunodeficiency syndrome, where leukemia was identified in 5 out of 20 patients. Nowadays, modern vectors have significantly improved safety profiles, but the risk remains theoretical for integrating vectors. Therefore, FDA currently recommends 15-year-old follow-up for gene therapy recipients due to these potential delayed effects. If we go to the next slide. And this, here we see the graph on the left that demonstrates the incidence of acute immune toxicities observed across pivotal large B-cell lymphoma CAR-T trials. So the cytokine release syndrome is the most frequent event, followed by ICANS, with majority of these being low grade and toxicity and manageable when promptly identified. The graph on the right provides valuable insight into the timing of these toxicities, and you can appreciate that relatively quick resolution of both cytokine release syndrome and neurotoxicity across different studies. These findings generated in the setting of rigorously conducted clinical trials where the patients are continuously monitored and managed by multidisciplinary teams with highly specialized expertise in cellular immunotherapy. This allows for rapid recognition and timely intervention with tocilizumab or corticosteroids, translating it into the favorable outcomes observed here. In routine clinical practice, where such intensive monitoring and immediate access to target interventions may not always be available, recognition and management can be delayed, increasing the risk of more severe manifestations. Therefore, this data highlight the necessity of treatment with experienced centers and education of the treating physician in the real-world setting. If we go to the next slide, please. And here we will look at the long-term effects. So, of course, the safety does not end when the patient leaves the hospital. CAR-T cell or cell therapy can persist for months to years, and depending on the target, antigen target, they can lead to the aplasia, cytopenia, prolonged cytopenia, B-cell aplasia, and hypogammaglobulinemia, all of which can increase the infection risk. We also must keep in mind and consider the theoretical possibility of the risk of secondary malignancies from insertional mutagenesis. The graph on the right summarizes some of the most relevant long-term adverse events observed following CAR-T therapy across multiple products. We see that the median follow-up duration for each study correlates with the longer, with the adverse effects that we see in the long-time follow-up. Of course, there are some key observations like high frequency of B-cell aplasia or persistent hypogammaglobulinemia, particularly with some products which help us to manage the patient and identify the ongoing infection prophylaxis and intravenous immunoglobulin replacement in some patients. Yeah, we also see a measurable rate of late severe cytopenia or late infections with some products, which also necessitate extended hematological monitoring beyond the acute phase. And yeah, overall, this figure illustrates that the management of CAR T-cell recipients does not end at discharge or at day 30. Long-term monitoring of cytopenia, immune reconstitution, infections, and secondary malignancies is an essential part of survivorship care. Can we go to the next slide? Yeah, long-term follow-up is particularly critical for advanced therapies due to their potential for persistent effects and delayed complications. For instance, in active clinical trial, we have the limitations because the typical phase 3 trial for advanced products includes only 50 to 200 patients compared to thousands for traditional pharmaceuticals. And this limits statistical power for detecting events with incidence rates below 1 to 3%. In pivotal ATMA advanced therapy trials, exclusion criteria typically eliminate 60 to 80% of real-world patients who would qualify for treatment based on their disease state, but are excluded due to comorbidities, age, organ function, etc. Most phase 3 advanced therapy trials have primary endpoints at 6 and 12 months with open label extension periods of 2 to 5 years. But potential concerns like insertional mutagenesis may take 5 to 15 years to manifest. So the key challenge is patient retention in long-term follow-up studies typically decreases by approximately 10 to 15% per year, highlighting the need for user-friendly monitoring approaches, close collaborations with the physician, treating physician, and maybe in the future digital health technologies. Next slide, please. Yes. It is very important to appreciate the difference between clinical trials data and real-world experience, as both provide complementary insights into the full impact of cell and gene therapy. The graph here illustrates this comparison very well. The orange bars represent the real-world cohort, where over 90% of patients were older than 65 years old, much older than the population enrolled in the pivotal trial. Despite this, 12 months survival remains very similar, showing the effectiveness of CAR T in routine practice. On the toxicity side, clinical trials show higher reported rates of cytokine release syndrome, cytopenia, and ICANS or neurotoxicity, partly because of standardized grading and close monitoring, while we know that real data often capture lower rates of acute events, but may provide insight into long-term effects, secondary malignancies, and comorbidity interactions. So this combination of data source ensures that we are not only validating efficacy, but also understanding how these therapies performs across the full patient population, guiding both patient selection and post-infusion care planning. If we go to the next slide. Yeah, real world challenges. So we do face challenges while implementing advanced therapies outside clinical trials because they introduce unique complexity that impact patient safety and data collection. So in the real world, patients are often older, have more comorbidities than those in pivotal trials, and this makes adverse event interpretation more challenging and increases the risk of confounders. data capture outside academic center is often fragmented, which is why registries and post-authorization safety studies are so critical to fill evidence gaps. And with this slide, I will hand over to Stephanie, who will talk about the operational experience and challenges in these long-term follow-up studies. Thank you.
Hello, everybody. On this initial slide, we wanted to start off with an example based on a product using the FDA requirements and the long-term follow-up requirements. This would be, for example, for something like a lentivirus product. As you can see in this scenario, we have a dedicated clinical long-term follow-up protocol. It is important that the investigators are prepared to maintain adequate and accurate case histories. As you can see, the first 5 years require a fairly robust follow-up as well that includes visits as well as physical exams, labs, etc. 5 years plus, you're looking at annual contact unless appropriate follow-up is needed as clinically indicated with test results. Just keep in mind, this is just an example of some of the FDA requirements, and other regions that use the EMA and other regulatory bodies will have different long-term follow-up specifics. And then also long-term follow-up requirements carry forward into the post-marketing phase through PASS and other registries. Next slide. So on this slide, we have a quick summary of the common product types as well as the long-term follow-up requirements per the FDA and EMA. As you can see, different agencies are more stringent on follow-up depending on the product type. For example, a herpes virus, we can have a follow-up of up to 15 years for the FDA, and then the EMA is at least annually until no risk is determined. Next slide. So an important piece when you're considering these long-term follow-up requirements is the shared burden across participants, sponsors, and sites. We need to be innovative and leverage technology in order to lessen this burden and make compliance with these requirements easier and the 15-year follow-up more sustainable. Retention of the participants as well as data quality is key. Planning for the long-term follow-up portion should begin in the very beginning of the study during protocol development and the startup portion of the study. When evaluating sites, we should ensure that they understand the expectations and requirements and can effectively relay this to participants so that they are informed before they start the study and they are ready to make the commitment. Over the next couple of slides, we will talk about some operational considerations as well as some strategies that can be used to support this burden and make it less for the participants, sites, and sponsors. Next slide. So in general, you want to design an operational solution that is fit for purpose to support the burden across the stakeholders. This slide is mainly focused on participant and site solutions. As an example of the consideration, is it the consideration of leveraging technology and patient-centric decentralized services to make it easier for the participants and sites. For example, using an application to do any electronic questionnaires for the patients versus having them come into the site, etc. Streamlining the protocol to minimize the visit frequency and leveraging standard of care visits, as well as existing data sources to collect safety and efficacy data should be considered to, again, lessen the burden on both the sites and the participants. Consider travel reimbursement services if remote visits, are not an option. And again, the overall key is to lessen the burden and ensure compliance with these long-term follow-up requirements. Next slide. On this slide, we map out a real-world scenario starting with screening and taking us all the way through long-term follow-up. As you can see in this example, we show that you can leverage both on-site clinic visits as well as remote visits. And then you would start off with the traditional CRA monitoring, perhaps starting with 100% SDV, and then moving to a centralized monitoring with remote data capture that would use key risk indicators to ensure that we are catching any outliers and determining what's going on with that site and really honing in to ensure data quality and compliance as well as patient safety. The goal is to keep participants and sites engaged and supported by leveraging various solutions such as e-consents, telehealth, eCOA, and home health services. Next slide. So slide 15, we're going to turn our focus to sponsor burden. This is an example of a potential solution that could be used for a sponsor that has similar products with the same data points that need to be collected. It's a streamlined approach where you would actually have all of the studies roll over into a long-term follow-up basket study, where we could, again, streamline everything for the participating sites, as well as keep the data collection and the safety monitoring all under one protocol to make it easier from a sponsor's perspective to analyze and run the analysis. Next slide. So overall, we are on the cusp of developing truly transformative cell and gene therapies. With innovation comes responsibility to adhere to the long-term follow-up requirements from the regulatory agencies to ensure participant safety and increase the confidence in these products. By designing flexible solutions, we can make long-term follow-up of 15 years sustainable. These operational foundations will also support the seamless transition into the post-marketing safety commitments. And with that, I will hand over to my colleague, Georgia. Thank you.
Thank you, Stephanie. So now that we have seen how long-term follow-up is managed in clinical trials, let's look at the legal framework that governs safety monitoring once a therapy is approved, so post-marketing. To begin with the EU, where ATMPs are the only therapeutic class with their own regulation. This is Regulation 1394/2007, and a guideline which is still under revision since 2018. This requires enhanced PV, including registries and long-term follow-up. FDA issued 2020 guidance for gene therapies, mandating up to 15 years of follow-up for some products due to delayed malignancies, mainly. UK follows more or less the same legislation as EU. Japan has a unique system, very specific, with conditional, time-limited approval for regenerative medicinal products, which is up to 7 years. I will not go through all the legislations. You can review them when you receive these slides, but what I would like to highlight is that while requirements different, the common denominator globally is the expectation for long-term safety monitoring, patient registries, and enhanced PV. And this makes harmonized global safety strategies essential. And we can see that science evolves very quickly, even faster sometimes than regulation, and this might be the reason why EMA guidance remains in draft, trying to gather more real-world data to make sure that the guidance will reflect practical experience. So this creates proactive PV approaches. Next slide, please. This is about the black triangle system. It is everyone is very familiar with that. It was introduced in the EU in 2012 to strengthen pharmacovigilance for new and complex medicines. For ATMPs, of course, it's automatic at approval because they are considered as novel biologicals with very limited data. The black triangle is legally required in the summary of product characteristics and patient information leaflets and all educational materials. The black triangle applies for at least 5 years, but for ATMPs, removal after 5 years is very rare because of the patient numbers, which remain small. Many adverse events may have delayed onset, such as secondary malignancies, and post-marketing evidence is still developing. So EMA and all national competent authorities use this black triangle to remind to all HCPs that every adverse event report matters. In ATMP, sometimes even a single case can trigger major regulatory action. And we have a recent example with EMA safety review in 2024 with CAR-T malignancies and secondary tumors. So the black triangle is not just a symbol, it's an active signal to physicians, clinicians, patients that real-world safety data is urgently needed. For ATMPs, the black triangle stays longer than the minimum period of 5 years, reflecting this complexity of these therapies. Next slide, please, Sonia. So here we have the sources of safety data. The spontaneous reporting is first. It remains the cornerstone of pharmacovigilance. As we said, for ATMPs, even a single spontaneous report can be significant because the treated population is very small. However, the spontaneous reporting is prone to underreporting, particularly for delayed toxicities. And this is the reason why we need solicited sources such as post-authorization safety studies, registries, and patient support programs. Solicited data can be structured and better suited to capture all these long-term outcomes. So for the post-authorization studies, they are usually mandated by EMA and FDA as part of the risk management plan. They can be interventional or non-interventional. Sorry, I hear my voice again. Then we have the registries. One good example is the EBMT, which is the European Society for Blood and Marrow Transplantation, which is very, very important for the CAR-T therapies because it enables structured collection of long-term outcomes across centers. Another program is the patient support programs, again, very well structured, which can generate valuable safety data. Then very important is the traceability of these complex therapies because ATMPs involve complex manufacturing and individualized production. So being able to link the specific product batch to a specific patient and their outcome is very critical. Because at that way we will be able to trace the product back through the manufacturing and delivery process to check whether any safety issues are the result of the quality issues. Next slide, please. Oh, here is the polling question. So, Sonia, if you could support on that.
Yes, I will, and I will launch it right now so everyone can participate. And there you go, everyone. You should see a blue screen take over. Now, I'll give you 45 seconds to complete the simple poll question. It's the only one we have for this webinar. And once I close the polls, I will take a look at the results and get right back into this great presentation. So the question is, what do you see as the biggest challenge in long-term follow-up post-approval? So please go ahead and select any of the options below. You have more than one option if you like. So choose all that apply. Is it patient retention over 10 to 15 years? Transition from trial to registry and pass? Or is it ensuring data quality in registries? And then the final option that we have for you to select from is regulatory changes and updates. So that's RMP, DHPCs, etc. So please go ahead, everyone, and cast your vote. Okay, that looks like it's half of our audience. I'm going to leave it open for a few more seconds here, Georgia, just to encourage everyone to participate, okay? Okay, that looks like it's the majority of our audience. So thank you very much, everyone, for participating in our poll question. I'm now going to close the polls. Okay, so Georgia, here are the results. 70% said patient retention over 10 to 15 years, then 30% for transition from trial to registry paths, then 50% for ensuring data quality in registries, and then another 50% for regulatory changes and updates. So thank you very much, everyone, for participating in poll question number 1 and our only poll question. Now, Georgia, before you get back into your discussion, what are your thoughts on the results of that poll question? Anything surprising there?
I think that's a very common perception with the 70% of patient retention, because keeping patient engaged for so many years is a massive challenge, especially when they have completed their treatment. And that is the reason why all companies, sponsors and pharma companies should invest in digital engagement tools and reminder systems and a good planning for all these transitions. But as you can see, there is no a single challenge. We saw 50% to the last two bullets, so everything is interconnected, and cross-functional approach is the solution on that.
Okay, thank you very much, Georgia. Now back to
Thank
you.
you. So project management in post-marketing PV, when a therapy transitions from the clinical trials to market approval, patients don't just fall off the radar. They should remain, as we said, under long-term safety monitoring. And regulators have different expectations depending on the regions. For example, in the United States, the FDA requires sponsors to follow patients under trial-based long-term follow-up protocols for up to 15 years. So FDA framework is still heavily rooted in trial-style follow-up. In Europe, on the other hand, we have a slightly different approach. Here, the responsibility sits more to post-marketing systems. So we are talking about registries and non-interventional safety studies, all of which are documented in the risk management plan. And there are, of course, other regulators such as PMDA for Japan and Health Canada that follow a mixed model, sometimes extending trial protocols, sometimes mandating registries. But from an operational standpoint, these regulatory differences have major implications for how we can manage them. For example, if the FDA trial protocol continues, but EMA requires a registry, you need a dual system running in parallel. So coordinating that requires strong communication, very good standard operating procedures, and clear role definitions across sponsors, CRO companies, marketing authorization holder sites, in order for this to be successful. Next slide, please. Long-term follow-up challenges and solution in post-marketing world more or less are the same, but with some distinct differences between the clinical trials. And the first challenge is patient retention. Unlike in clinical trials, here we don't have frequent site visit or sponsor follow-ups once the therapy is authorized. Patients may feel finished with treatment or disengaged from safety monitoring. To address this, we emphasize retention from the very beginning. So educating patients during treatment that follow-up is part of their care journey. We also use, as we said, digital tools such as apps, SMS reminders, patient portals to keep engagement high over their years. The second challenge is the transition point when a therapy moves from trial, for example, to authorization, and patients have to be transferred into a registry or a post-authorization safety study. Without careful coordination, again, data and patients can be easily lost. The solution on that is structured handover, ensuring that patient consents cover registry participation, harmonizing forms and data elements between the trial and the registry, and setting up centralized tracking system that allow us to follow each patient from one system into another. Another challenge is the variability of participating centers. Registry reporting is so often voluntary and less resource treating physician may see registry entry as administrative work on top of all these clinical duties that they have. Staff turnover also makes continuity harder. To overcome this, we have to make processes as simple as possible for the clinicians, streamline forms, clear against standard operating procedures, and alignment with routine visits, so data entry is not an extra work. Continuous training for new staff is also essential to keep data quality consistent. Post-marketing studies are global, and each country may have different registry structures or reporting requirements or even data privacy laws. And this creates complexity for sponsor and marketing authorization holders. We address this through centralized coordination, harmonizing data fields across the registries, ensuring compliance with regional requirements, and creating global oversight systems that bring fragmented inputs in one coherent data set. Data quality is another challenge. Unlike again trials that CRFs are strictly monitored, registry data depends heavily on what centers choose to report. So we have to define a minimum essential data set aligned with regulatory expectations, push for more electronic health records integration where possible in order to reduce manual entry and monitor the data quality. Last is the rapid regulatory updates, which is a very big challenge. And to overcome this, we have to coordinate the cross-functional teams, pharmacovigilance, marketing, medical, in order to ensure that all the materials are rolled out efficiently and to track whether these measures are actually effective in changing practice. But the real challenge in post-marketing isn't just keeping patients engaged and registries running, but the data that we collect should be complete and reliable. Because if patients are lost or data is inconsistent, our ability to detect new safety issues is compromised. And that's where signal detection comes in. And I will hand over to Ian, who will explain how we can use this long-term data to identify signals.
Thank you, Georgia. So when we talk about signal detection in advanced therapies, I think the first point to emphasize is just how different the landscape looks compared to traditional drugs or biologics. For most marketed medicines, we build our understanding of risk on the back of many thousands of exposed patients. This gives us statistical power to use disproportionality analysis or other quantitative data mining approaches. By contrast, many cell and gene therapies were approved with fewer than a couple of hundred patients exposed and across highly selective trial populations. And really that means that the statistical tools that we usually rely upon are essentially ineffective. The second challenge is the nature of the risks themselves. These are novel products. They're frequently designed to trigger profound immune or genetic changes. And as we've heard, that introduces quite unique acute toxicities, cytokine release syndrome, immune effector cell associated neurotoxicity, and other inflammatory syndromes that could be life-threatening. And equally concerning, as we've heard, are the delayed toxicities, such as insertional mutagenesis and secondary malignancies that may not even appear until years later. So these risks are fundamentally different from what we deal with in conventional pharmacovigilance. And it's really for that reason that we've got the regulators at mandating the long-term follow-up. And given that most serious events may not occur during clinical trials or early after post-marketing exposure, patients, of course, need to be followed up for a decade or more before the full safety profile can really be characterized. And finally, I think the safety data that comes from the long-term follow-up is fragmented and it's often incomplete. We've got data from spontaneous reports, we've got past studies, we've got long-term safety data available through registries and published literature as well. And these data sets, they're often siloed. They may be incomplete in themselves, of course, subject to underreporting, and that's a well-recognized problem in post-marketing pharmacovigilance anyway. So put together, these factors create a kind of really unique and difficult environment for signal detection, and every individual case really carries a disproportionate importance. Next slide, please. So the backbone of pharmacovigilance for advanced therapies does remain the spontaneous and solicited reporting. While we've got the underreporting as a recognised limitation, even a single well-documented case for these products can generate a signal, particularly if it highlights a novel risk such as a secondary malignancy or an unusual inflammatory presentation. But data itself alone is not sufficient, because it's the interpretation that's important here. It requires expert clinical adjudication, cross-functional safety committees that bring together not only the pharmacovigilance professionals, the clinicians, but also the regulatory experts, and others even, to ensure that we can really truly assess the plausibility and the context of the event. And this qualitative multidisciplinary approach is just as important as any quantitative signal detection tools used in other products. Another critical component is the traceability. For such therapies or products that are manufactured in small patient-specific batches, the ability to link a product or batch to a patient outcome is essential. So without robust traceability systems, the delayed adverse events that can occur years later after the infusion can't be properly attributed. And then finally, the structured registries that are mandated by the regulators, they really provide the foundation for the long-term follow-up and the long-term safety profile characterization. These registries allow systematic capture of safety information and patient outcomes far beyond what's possible in clinical trials, of course. And in practice, they do form the foundation for the long-term profile monitoring. Next slide, please. Artificial intelligence, a topic we're all hearing a lot about, but also one of the most powerful contributions of this in pharmacovigilance is going to be its ability to bring fragmented data together and analyze that data. So with our safety information today being dispersed, AI has the ability to harmonize the information, and it's got the ability to recognize when weak signals across different sources might all be pointing in the same direction. And this then gives us a consolidated view of risk that might be impossible to achieve manually. Once we've got the data integrated, AI can also enable multidimensional pattern recognition. So rather than looking at individual case counts in isolation, algorithms can explore associations across patient subgroups, product platforms, vector types, indication, and even comorbidities. So for example, AI might identify that a neurotoxicity signal is emerging only in a particular CAR-T construct, specifically among patients with prior CNS disease and treated for a certain tumor type. And that level of granularity is extremely difficult to uncover through manual review alone. The third major strength is early signal amplification and risk prediction. So traditional quantitative methods rely upon statistical threshold having been crossed before a potential signal is flagged. AI can detect patterns while they're still weak, so they can be evaluated much earlier. And beyond that, predictive modeling allows us to anticipate where risk is likely to occur in the future. So for example, registry data could suggest that younger patients with a certain genetic background or patients with pre-existing cardiac comorbidities might be more vulnerable to severe CRS or long-term complications. And this then moves us from a reactive model of pharmacovigilance to a proactive anticipatory one. And of course, regulators are clear that AI must remain a supportive tool. Both the EMA and the FDA stress that algorithms must be transparent, outputs must be auditable, and all findings subject to human adjudication. And in practice, that means that AI can support with integrating complex data sets and highlighting patterns far earlier than a human could, but the responsibility for interpreting and acting upon those findings remains firmly within the realms of the pharmacovigilance and the clinical experts. Next slide, please. So I'd like to hand back over now to Georgia to talk about risk management in the context of the risk management plan.
Thank you, Ian. From a regulatory perspective, every ATMP must have a risk management plan in place from day zero This plan isn't static. It evolves as new safety data emerges over time. Regulators expect risk management plans to include several key elements, all these that we have already discussed. Long-term follow-up, often through post-authorization safety studies and registries, educational materials tailored for both healthcare professionals and patients, controlled distribution systems in order to ensure that the product is used safely and correctly, and communication tools like DHCCs to share urgent safety information rapidly. But it's not just about listing these tools. Authorities require evidence that they are actually working. So measuring the effectiveness is very crucial. And just one last point, and while we're not focusing only on the EU today, it's hard not to mention that the role of the QPPV and the PSMF on all this. And as a QPPV myself, I'm deeply involved in ensuring that these activities are not just theoretical, but actually implemented and monitored and very well documented. One of these supported documentation are pharmacovigilance system master file. And although the regulations do not require a separate PSMF per product, for complex therapies like this, like ATMPs, it's often very helpful to maintain a dedicated section or module within the PSMF. But even with the right system and governance in place, there is also one area where implementation is always complex, and that it's at the clinical level. Making sure that these tools are actually usable and practical for physicians and patients is a real test. So as we conclude, it's critical to emphasize 4 points. First of all, it's the enhanced clinical monitoring, which is not optional, it's very essential, and CGTs demand very sophisticated therapy-specific protocols that evolve with our understanding and real-world experience. Second, we have the specialized medical expertise, which is irreplaceable. These presentations involve complex and unpredictable safety profiles that require the physicians who understand both the therapy mechanism and the patient's individual risk factors. Third, we need integrated data systems that capture real-world outcomes across different populations. Our current fragmented approach leaves dangerous knowledge gaps, and important safety signals may be missed. Finally, education is critical. We must train all physicians to recognize, evaluate, and manage the long-term consequences of these revolutionary therapies. The success of cell and gene therapies depend not just on their efficacy, but on our ability to monitor and manage their long-term effects through proactive pharmacovigilance. This is our responsibility for all the patients and the future of these medicines. Thank you, and I hand over to you, Sonia.
We have time for just maybe one or two questions, so let's get into that right now. The first question I have here is, why do regulators insist on effectiveness checks for educational tools and DHPCs and ATMPs? Who would like to take that question?
I can take that, Sonia. Simply, sending information doesn't guarantee that it's understood or used properly, especially for such complex therapies like CAR-T or gene therapies. Physicians must be actively engaged. So it's not enough, to drop just a letter in the email. These tools have to be delivered through structured and organized channels that ensure that physicians actually receive them, they read them, and they can apply them in practice. And this requires cooperation between PV, between pharmacovigilance experts, local competent authorities, national health systems, in order to create reliable distribution frameworks. So a coordinated effort is very crucial.
Mm-hmm. Okay, I'm going to squeeze one more question in. This audience member is asking, why is long-term immune surveillance critical after CAR T cell or other CGTs, and what biomarkers are most informative in predicting late toxicities or relapse? Who would like to take that question? It's a great question.
I think this is I would take this question. I think this is long-term immune surveillance is quite crucial because, as we mentioned, the CAR T cell persistence is strongly correlated with durable remission in hematological malignancies, but also increases the risk of late cytokine release syndrome or immune effector cell-associated neurotoxicity syndrome. Then we follow immune reconstitution. The cell and gene therapy can cause prolonged cytopenias or even aplasia, requiring tracking of immune recovery to prevent opportunistic infections, and of course the risk of relapse. So early detection of clonal evolution or antigen escape can give us the idea or guide us preemptive interventions and to the examination of patients. Regarding the key biomarkers, it is in CAR T cell levels, the qualitative PCR or flow cytometry, persistence, and the exhaustion markers, the PD-1, LEG3 expression, etc. But it heavily depends on the products that we use.
Okay. Well, thank you very much for answering those questions. We have reached the end of the Q&A portion of the webinar. For those of you who are interested in hearing more from Allucent, please utilize this information here. They have another upcoming webinar that's slated for Wednesday, October the 29th, Smarter Site Feasibility Strategies for Predictable Enrollment in Late-Stage Oncology Trials. So I put the link in the chat box there for everyone to utilize, and you can also register right here with this information. Okay. Now, if we couldn't attend to all your questions, and there's quite a few of them, remember the team will follow up with you after this presentation. And if you have any further questions, please utilize the email addresses you see there on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, I have shared a link to view the recording of today's event in the chat box along with the next webinar for you to register, so please go ahead and utilize that. And for those of you watching on LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. And get even more out of today's presentation by downloading the supporting material that we have there for you, a PDF, Drug Safety Pharmacovigilance. So please go ahead and check that out too. Now please join us in thanking our speakers for their time here today. So thank you everyone for being here. Georgia, Stephanie, Ian, and Oksana. Thank you so much. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until we see each other again, please take care. Bye for now. I'm Sonia Hunt. Bye, everyone.